chapter 3, tools of the laboratory, how we do things, how we identify, how we look at things in microscopes, and how we determine what's what in microbiology. Okay, first thing we do is to identify the microorganisms. If we're going to have a chance at understanding what's going on, either your patient or with pandemics, we have to identify what microorganism is that's causing it. So one goal of these procedures is to attach a name or identity to the microbe. We have to be able to name it so we know what we're dealing with. We use all the information that we gather from inspections and investigation. Identification is accomplished through the use of keys, charts, computer programs that analyze the data and arrive at a final conclusion. Okay, we have to inoculate what we're dealing with. We have to introduce the virus into a virus or the bacteria or the pathogen in a sense so we can study it. And we do that through several different, we use different media. A medium is just something that we can basically grow either the virus or the pathogen bacteria in that. Okay. So the medium has to support the growth of what we're dealing with, the microorganism. Okay. It could be a solid, a liquid, it could be held in tubes, on plates, flasks, or even in eggs. Delivery tool is either a loop, like we'll do loops when we streak an agar plate. We'll put bacteria on there and use that to go back and forth to streak it. It'll be like a needle, so you can inject it like into a bird embryo. Okay. A swab or a syringe. You can drop it in like a little. Sit down. Okay. And then we take information we get about that microorganism. Okay. Additional tenets for microbial functions and characteristics may include inoculations, okay, into specialized media that determine biochemical traits, immunological and immunological testing, and genetic typing. So depending on what we want to do, whether it matches DNA, test its sensitivity, test its biochemical reactivity, okay, that depends on the media we inject into it, or media we place that pathogen or that microbe onto. Okay. We collect specimens of it. Okay. Microbiology is being by sampling object of their interest. Okay. Very common sources that we use to sample body fluids. We go urine test for the doctor, blood tests. Okay. Foods, water, soil, plants, animals, even places like icebergs, volcanoes, rocks. We can all take samples of that and analyze those for different microbes that occur naturally in those things. And once you get the sample, we incubate it. We have to have it grow. We have to put it in an environment that's conducive to it growing. Okay. So inoculated media, once it's been put into the media, whether it's a solid or liquid, they are placed in a controlled environment. Culture develops, you get a visible growth of the microbes in the container. You'll get fuzzy, you'll see the agar plates where you'll get this where you put the streaks in there. It will show you how the microbe as it's growing. And then you look at it. Once you grow it, isolate it, then you look at it. You observe, you have a macroscopic appearance, which is what you can see on the fuzzy plate, the fuzzy circles or square, however it develops itself. The colonies is what you look at macroscopically. You don't need a microscope for that. Then you examine under a microscope. On the microscope, for basic D cell type, you see the shape of the different cells, whether they're cocci, which are round, or bacilli, or rod-shaped. This may be enhanced through staining and the use of specialized microscopes. Some inoculations to separate microbes create isolated colonies. So you can either isolate them to the different species of they have different microorganisms. So that you isolate each contains a specific or a single type of microbe. This is invaluable for identifying the exact species of a microbe. You just isolate it first in a sample. Okay. So this makes you have pure cultures. So you don't get any contamination from a different microbe, which is really important. I mean, say you're dealing with a patient that has a disease, okay? You isolate that microbe. If it's contaminated with a different microbe, it's going to react differently to medication when you test it. So it's real important to get a pure culture when you're testing for like antibiotic sensitivities and things like that. So the microscope has two key characteristics. What it does, it's magnification, okay, and resolving power. I mean, it does no good if you make it big enough and it's all blurry, right? You got to do both. You got to make it bigger and you got to make it so you can see the details so you can focus on it. So microscope has magnification and resolving power. So making it bigger so you can see it, and making so you can see the details of it. It's not just fuzzy. So magnification most results from interaction between visible light and the curvature of a lens. The extent of the enlargement is the magnification, and it magnifies things by the curvature of the lens. This is the different parts of the microscope. You have your ocular lenses, which is what you look through, and it's also a magnification process there. You got your objective lenses, which is also magnification. So there's two magnification parts, the objective lens and the ocular lens. Both have magnifications to those. You got a revolving nose piece, so you can select different lenses, different magnifications. You have a condenser for condensing and putting light into a specific column, so it goes through individually. You have a diaphragm that helps focus the light. You have the light source to shine light through the specimen. You have a coarse and a fine focus. The coarse brings it into rough focus. The fine focus focuses each individual aspect of it. You have a voltage control for the lamp to determine the light intensity. And you got a mechanical stage that you move around. You can move it left to right, right to left, to move around through your specimen. Those are the parts of the microscope. So we said magnification has two phases. The objective lens forms a magnified real image. Real image is projected to the ocular lens, where you're looking through, where it's magnified for the virtual image. So there's two areas where it's magnified, and both have to be taken into effect. Okay? You got your objective lens, and then you got your ocular lens. Here's the deal. Here's your light source. Comes through the diaphragm, and it condenses light to focus on your object. And here's your objective lens. This is the first magnification step right here. And it flips your real image around, reverses light. And then you have your ocular lens where you're looking through. Okay, this is your eye, look down through here. And your ocular lens is your second magnification step. With a 1.5 or a 2 for that one. So total magnification, okay, of your final image is a product of a separate magnified candidate of the two lenses. So both lenses contribute to magnifying your specimen. So the objective power times your ocular power gives you your total magnification of what you're looking at. So remember, your objective power is at the lens of the microscope. Your ocular power is at the ocular where your eye is focusing on. Total magnification, again, the same picture that we've had. Total magnification is your light source. Sit down, Michael. Okay, so as we did before, condensation of it. So your objective lens provides your real image. Your ocular lens provides your virtual image. It's a product of those two for your total magnification. So when we talk about resolution, you have to define, you have to be able to see, not just magnify it, but you have to be able to see it in great detail. Okay. So the capacity to distinguish and separate two adjacent objects depends on the wavelength of light that you're using, which hits the image, and the characteristics of your objectives, or what kind of media it is. Listen now. Okay. All right. So once you're quantifying resolution is your resolving power, which is your wavelength of light in nanometers. Remember, nanometers is what you usually measure light in, and I use like 400 to 750 is your visible light range in nanometers. And nano means 10 to the minus 9 meters. Very small wavelengths of light. So your resolving power of your microscope is your wavelength of light divided by 2 times the numerical aperture of your objective lens. So yeah, visible light is right here, between 400 and 750 nanometers. That's the range of visible light. The numerical aperture of the lens is used between 0.1 to 1.25. That's where you put your eye to, that part right there. So the shorter the wavelength and a larger numerical aperture will provide a better resolution. When you think about it, if you're looking at an object, shorter wavelength, if your wavelength is like this here in your object, it's going to be very broad area. If your wavelength is extremely fast, it's going to hit much more times. And you get a much greater resolution with the shorter wavelength. Much better image quality with shorter wavelength light. You can also use oil immersion. Oil immersion resolution is 0.2 micrometers. A micrometer is 10 to the minus 6. That's a much shorter wavelength. So magnification of your lenses between 40, we usually start, sometimes 10, we usually 40 to 2000. What the oil does, the oil cuts out the air between the lens and your subject, what you're looking at. Makes you able to focus a lot better, much higher resolution when you use oils, oil immersion. So here's a oil. This is your objective lens you're looking through. Here's the curvature. Okay? And you can see as it passes through air, comes out here and deflects through the air. When you have oil here between your lens and object, there's no air. There's no dispersion of the rays of light. They don't bounce anywhere. They go through the oil, and the oil directs them straight into the objective lens. See, all the focus comes and stays in the field of vision. So you're not getting the scatter out here to the side we do with air. We're hitting the air molecules. And the oil focuses that, makes it a much higher resolution. So you can use a higher magnification lens because you got better resolution. Okay. You can use the same magnification lens in air, but the light scattering, so you can't see it as well. It doesn't give you the detail. With oil there, you increase your resolution, increase your detail of what you're looking at. So this is just an example of what you can see. Okay. This is the different types of bacteria you can look at with the different magnifications of it. See how this is like times 1,000 across down here. And you can see the different virus particles, different bacilli, the flagellum, things like that, as you're looking through the microscope. So in order to get the best resolution possible with a light microscope, you want long wavelengths, short wavelengths, or the wavelength doesn't affect resolution. Which wavelength do you want?
Short wavelengths?
Want short wavelength because it hits it so many more times, it makes the resolution much greater than if you're sending off wavelengths of a longer wavelength, a light of a longer wavelength. So it should be B, right? Yep, shorter wavelengths of light. Perfect. All right, variation on the optical microscope. Any questions so far?
No, thank you.
Perfect, no problem. Okay, we can do the different variations on our microscopes. We can use a bright field in the back. So that you see the background here is white. It's a bright field in the back. It's widely used for specimens that's darker than the surrounding field. It's used for live and preserved stained specimens. You can do a dark field where the background is black. Okay? It's darker than your specimen. Brightly luminous specimen is surrounded by dark field, used for live and unstained specimens. You do a phase contrast. It transforms subtle changes in light waves passing the specimen through differences in light, different light intensity. Best observed for intracellular structures. We got Janice coming on too. We got another person. All right. And then we also have fluorescence microscopes. Okay, that you have something that glows like a red tide at the beach. We have those the microscopes that often they glow in fluorescent images. So modified microscope with ultraviolet radiation sources and filters. It uses dyes that emit visible light when bombarded with shorter UV rays, which give off fluor. It's useful in diagnosis and in diagnosing infections. But we just lost that last person too. Scanning, confocal microscope. This uses a laser beam. Laser beams are very specific wavelengths of light. Use laser beam to scan the specimen to go across it. This integrates the image to focus on multiple depths or planes. And then you can focus on the cilia on the area or cilia on top. You can cut it in different lanes. Like doing surgery with lasers, when we do brain surgery, we use a laser to focus at different depths. We don't have to cut through the skull anymore. We use lasers to do that. They'll only cut at a certain depth. That's what you use after the microscope also. It'll only focus at a certain depth as you take a three-dimensional specimen. You can see the cilia on the outside or the cilia down deeper as you determine the depth of the laser. Electron microscopy forms energy of the beam of electrons. They can be to travel in a wave-like pattern when accelerated at very high speeds. Electron waves are 100,000 times shorter than visible light that we use. So much shorter wavelengths, much more magnification because it gets much higher resolution. Electrons have tremendous power to resolve minute structures. Resolving power is a function of wavelength. Magnification is between 5,000 and 1 million times. So much greater than a light microscope, because the electrons have a much shorter wavelength and they can hit those little areas and much more accurate. So this is just a summary we've been talking about between the light microscope and electron or transmission microscopes. Okay. You see all the different characteristics of each one. We have different types. You have two types of electron microscopes: scan electron microscope and transition electron microscopes. So the TEM, the transition electron microscopes, transmit electrons through the specimen. And these darker areas represent thicker, denser parts. Lighter areas more transparent, less dense, because more electrons travel through. If there's less resistance, more electrons are going to travel through, giving you a lighter field on your medium. Scan electron microscopes provide detailed three-dimensional views. Okay. SEMs, or scan electron microscopy, bombard surfaces, okay, of a whole metal-coated specimen with electrons while scanning back and forth over. Specimen preparations for an optical microscope. We can have different wet mounts and hanging dry mounts. Those are examination characteristics of live cells. We can check their size, motility, how they're moving, their shape, and the arrangement of the cells in the specimen. Okay. Those are wet mounts so they can move around. Like you put a drop of pond water, that's a wet mount, because things can move and you can tell how they interact with the medium. Fixed mounts, they're not motion, there's no motion involved. Fixed mounts made by drying and heating of film. Okay. The smear is stained using dyes to visualization of certain cells or certain cell parts. So we're dyeing certain areas once they've been fixed to determine and look at individual parts of the cell. So staining, you can stain everything. Dyes are used to create contrast by giving it color. Basic dyes, okay, are cation, ground a positive charge. They're positively charged chromatophores. So you have ions like in muscle cells, like calcium 2+. You can use a basic dye to bond to the positive charge. The positive staining surfaces of the microbes are negatively charged and attract your basic dyes. Okay. Acidic dyes are anionic. They're negatively charged chromophores. They check the negative charges, like the OH- hydroxide ion. Negative staining microbes repels dyes. The dye stains the background. So here you see the background is completely stained, and it gives you good visualization of the cell that's in it as you increase magnification. So you can do negative staining, which stains the background. Simple stains. Simple stains is one dye is used. This reveals your shapes, your size, and your arrangement. Differential staining. This is a primary stain and a counter stain. So it distinguishes different cell types or parts, like a gram stain, acid-fast stain, and endospore stain. So you can have a specimen and you can stain the different parts individually to isolate each section you want to look at, so you can see it very clearly. Or you can have structural stains. Structural stains reveal certain cell parts. You can stain mitochondria. You can stain the cytoskeleton of the cell. You can stain the Golgi apparatus. So you can see the different aspects of it. So you stain the structures. And this will train you you have the capsular and flagellar stains. You can have simple and negative stains. Use one dye to observe the cells. Here you can see the cells are what are stained. This is a crystal violet stain of Staphylococcus aureus, and this is 1,000 times magnification. You see the round shape of each individual micro there. Okay, this is the same thing. You see the rods that are stained and the individual cocci that are stained. So here you have the Bacillus and Staphylococcus made with nigrosin, and this is also 1,000 times magnification. You have differential stains, like we talked about. Differential stains stain different aspects of it. Use two dyes to distinguish between two different cell types. You have the gram stains. You get purple cells are gram positive. You have the gram stain on it. The red cells are gram negative stains that they pick up. So this picks up your positive stains, this picks up your negative stains. So it allows you to differentiate between gram-positive bacteria and gram-negative bacteria. They respond differently to different bacterial treatment, antibiotic treatments. So it's important to determine whether it's gram-positive or gram-negative of what you're treating in your patient. This is acid-fast stains. Okay, the red cells are acid-fast. The blue cells are non-acid-fast. Difference between different cell types of what you're looking at and what your patient has in their body. You can have spore stains. Spore stains just show up these little green areas right in here. And vegetable cells are the red ones. Okay, this is also 1,000 times. And these spores are extremely hard, are much harder to kill. The little spores right here, you can kill bacteria, antibiotics, you can usually with heat. But autoclaves, like in a dental office, okay, they have to have under a certain pressure and a certain heat to kill everything. So these spores or the endospores, okay, don't die as easily as the bacteria do or the viruses. So what we do is we have to run a test every week on our sterilization equipment. And inside it, we put this little envelope inside the sterilizer, along with our instruments and stuff. We run it. And then we take this envelope, which has these spores, live spores inside the envelope. It's a sealed envelope. Then we send that sealed envelope to the CDC. They analyze it and make sure those endospores are killed. If you killed the endospores, you've killed everything else, because those are the hardest to kill. Those are not killed just by heat. You have to have heat and pressure for 20 minutes to saturate them and kills those out. And you have to do it once a week for any medical office to make sure your sterilizer is working. So we use these endospores to determine that, because they're much harder to kill. The structural stains we talked about, where you can have the different structures inside the cell, used to enhance cell details. You can enhance what's inside the cell. This is a capsule stain of Rajnaid bacterium 1,000 times. And you can see inside each individual cells what's being stained there. To magnify that. Flagellar stains of Bacillus cereus. They have a fine fringe of the flagella. So a little flagella right in here. And you see how the flagella stain along with this. And this is what makes those bacteria mobile. Okay? It's like the flagella, like a flagella on a sperm that allows it to swim through up into the egg. Okay? These are bacteria there's two purposes of these flagellum around the site. One is to change the position of the bacterium so it finds a different food source. If you're in a certain environment, you know that food, you got to move to another environment. So it allows bacteria to move. The second thing it allows it to do is to move the bacteria to stay stationary and move the solution around it. So if these things beat in a direction and push this fluid away, more fluid is going to come fill its place and more nutrients come into it. So there's two functions: to actually physically move the bacteria to a new location, and to move and bring more nutrients into the same area the bacteria is in. You can really clear all the little flagella there. I mean, I'll take my little lines away, but the flagella you see really clearly marked in that stain. There's 6 I's of culturing microbes. There's inoculation. Inoculation we talked about at the beginning, where you introduce the sample into the container to produce a culture. You do it by swabs. You can do it by injection. You can do it by taking a sample from chipping off a piece of rock or a piece of substance. There's all kinds of introducing a sample into the container. Then we have isolation. You want to separate one species from another. Like I said, if you have a patient and you have multiple microbes on your sample, which one do you treat for? You have to isolate the microbes and find out which one is doing what in the body. Incubation. Incubation occurs in a solid or liquid medium, and it allows growth. You put it in an environment that helps the growth of bacteria or the pathogen. Then you inspect it. You look at it. See what it looks like. You look at it macroscopically to see what kind of colonies it forms, and look at it microscopically to see what the shape it is, see what type bacterium it is. Then you gather information on that bacterium or that microbe, and then that leads to identification. Identification is the key. That's what you want to do. You want to identify what's infecting your patient. You want to identify that microbe. I think one of your labs is you get an unknown and you get to identify the microbe. So we had inoculation, so we have an isolation. So we're going to isolate the different types, so they have different growth on the media plates. You get different samples. So if an individual bacteria cell is separate from other cells and has space of a nutrient surface, it will grow into a mound, which is called a colony. You can see these are colonies right here as they grow. Each colony consists of only one species. You can streak the plates we talked about, where you take your loop, you put in your agar plate, and you go back and forth along the plate. And you'll do it overlapping different directions. Help isolate your colony. This works best if the spreading tool used the inoculation loop is re-sterilized after each step. So your loop is basically a little wire loop on the end of the stick, and you dip this into your specimen, your media, and then you spray it, or not spray it, you spread it along the plate. After each time you burn it, you set it to a flame to sterilize it and dip it again between each step. And this is what your plate's going to end up looking like. And you can see the different colonies that are forming, that are isolating the different types of bacteria or different types of microbes. You have the pour plate technique, which is it allows you to purify your sample technique. In other words, you put your loop in your container here, do your species, put that into there, dip your loop again, into here, dip your loop again. So each time you're getting purer and purer modifications of your agar plate. You dip it in, grow it, take a sample from there, sterilize it, dip it in, grow it, take a sample from there, dip it in, grow it, and you're just diluting it until you get a very specific one species in each colony, and that's what you want. It's also called serial dilution, because you dilute your specimen over and over and over until it gets to the point where you have pure specimen. The spread tape technique, take your honey stick, you spread over the plate, and you let it grow in the different agar media. That's a simple technique. Just put it in, let it grow. After you get it, you do your inspection. Like I said, you look at it macroscopically and microscopically. If a single species is growing in the container, you have a pure culture. If there are multiple species, then you have a mixed culture. The idea is to have a pure culture. You check for contaminants in your culture if you don't have a pure culture. You have to know what's contaminated. So you look at it, you can see specific cultures, pure cultures. You can do it on a solid media. You can do it in a liquid media. See it growing there, growing there. So you can see your microbe growing into different mediums. How you identify a microbe? You take the cell and the colony morphology and staining characteristics. What stain does it absorb? Different microbes absorb different stains. So we take the morphology, the shape. Different colonies take on different shapes. Some have a halo ring around it. Some will be spherical. Some will be not rectangular, but ovoid. And they have DNA sequencing. And each microbe will have different colonies of different DNA sequences, if they're different colonies of the microbe. You have biochemical tests that are organ its chemical and metabolic characteristic, how it metabolizes nutrients. And you've got immunological tests, like we do for blood type. A, B, and AB are called different immunological tests of our blood cells. So media we talked about. Okay. Media can classify according to three properties: what you put your bacteria or your microbe in. The physical state is either liquid, semi-solid, or solid. The chemical composition of the media is going to be synthetic, or chemically defined by man-made, or complex. Steps in it. And the functional type can be a general purpose, can be enriched, can be selective, differential, anaerobic, transport, assay, enumeration. All depends on what you want to do. You can have different functional types of media, how you want it to respond. Certain bacteria only grow in a no oxygen part. Chat in here too. Let me check that. I'm just having a hard time. Okay. All right. And physical states of the media could be a broth. Okay. Broths do not solidify; they're liquid, and a broth is just a nutrient-based liquid. Okay. You can have a semi-solid. Okay. It contains solidifying agents that will solidify later on, or you have a solid, which is the firm surface we call it. So these solids contain solidifying agents to create a solid media for them. They're liquefiable and non-liquefiable. Some can be heated, like agar plates can be heated and turned into liquid and poured into plates. Some are non-liquefiable. You cannot heat and turn them into liquid. So here's your liquid state. You add your bacteria into it. You see your microbes growing in the liquid here in this plate. You can see it in this plate also here too. The most common used solidifying agent is agar. The solid at room temperature. It liquefies at boiling water. Does not re-solidify until it cools to about 42 degrees centigrade. This is above room temperature. Provides a framework to hold moisture and nutrients in place so bacteria can grow and expand. And it is not digestible by microbes. So you're not going to destroy it as the microbes feed on the nutrients inside the agar. The agar is not going to be dissolved by the microbes. So you boil it on a hot plate, you pour it into your petri dish, you seal it over, and it solidifies as it cools down. You take, and then once it solidifies, you take your wand, dip it in, and then you do your streaking technique of your different microbes on your agar plate. All right, we have the nutrient broth. That's your liquid medium that contains beef extract and peptone. I mean, you're familiar with broth, chicken broth, vegetable broth, beef broth. It's the same thing. It's just a liquid medium that has nutrients in it to support the growth of the microbes. And the nutrient agar is a solid media containing beef extract, peptone, and agar.
What do you mean solid media? Like it's like a almost like the collagen of the beef extract? Like the
Yeah, it solidifies. It's like the fat. Like the agar plates, are you boil them and then they're liquid, then you can pour them into a plate, let it sit, and it will solidify. They become a solid, which you can put streaks on. The liquid media just maintain liquid.
Thank you.
Yeah, if you saw, I'm trying to think of the agar plate's like a thick gel consistency. They won't spread if you tip it upside down. They'll stay in there, but they're still like pliable on top. The bacteria adhere to it really easily. That's a solid media versus the liquid media that you pour into a test tube. That make sense?
Yes.
Perfect. So chemical contents of the media. The synthetic we talked about, it contains pure organic and inorganic compounds in an exact chemical form. So depending on what you want to make, we synthesize it as synthetics. We make it in the lab. We add the different materials that we need, put them together, put them into a binding agent, and we can solidify it. Complex or non-synthetic contains at least one ingredient that's not chemically defined. We didn't make it in the lab. It's a natural occurring component that the bacteria can use as nutritional. General purpose media grows a broad range of microbes. This is usually non-synthetic. This is your agar plates with the nutrients in there, and the microbes can grow on that plate. Enriched media. Enriched media, we add a whole bunch of stuff to it to help the microbes grow. Contains complex organic substances like blood, serum, hemoglobin, special growth factors that are required by very specific or fastidious microbes. Certain microbes only respond to certain types of media, like hemoglobin. So we get media, a hemoglobin-rich media that's called an enriched media, or blood serum. This shows you growing in a blood serum. This is beta hemolysis. You can see the bacteria or the pathogen, the microbe here, and around the microbe, you see that white ring? That's where the pathogen is destroying the red blood cells around it. Hemolysis, causing lysis of the red blood cells. Selective media contains one or more agents that inhibit growth of some microbes and encourage growth of other ones. So, selective media The media is only going to allow a certain microbe to develop. Otherwise, it's going to die off. Differential media allows growth of several types of microbes and displays visual differences among these different types of microbes. See the difference between. So selective, we kill off some of the ones we don't want. Differential means you allow several types to grow. So Crow-Mager contains several dyes and is used to diagnose urinary tract infections. Patient sample is inoculated, and based on the color of the colonies, you can identify the pathogen. Crow-Mager is best described as what type of medium. It's either enriched, we put it in, selective, differential, or complex.
Is it complex?
Complex would be.
Or is it selective?
Maybe differential. Yeah, differential.
Oh.
Differential media because it allows different. You can see how it grows this one, and then it grows E. coli, and Klebsiella, and Proteus enter. And you see the different colors that it grows, the different bacteria. So you can streak it on there with the different bacteria. It's a differential media, allow different types of bacteria to grow in that same medium. Now, complex is also a differential medium, and I wouldn't mark that wrong, but differential is a little more correct. Does that make sense? In a complex media, you have the same thing, you have differential growth, but it's a more of a complex situation where the composition of it. This is used to diagnose urinary tract infections. Let's just look for a specific type. So that would be a differential media. Does that make sense?
Yeah, so yeah, it does, because it's showing you the different bacteria specific, but within the same media.
Correct. Perfect.
Yeah.
Let me explain it. Yeah. So some media can be selective and differential. Differential allows different types to grow, but it's selective for only those three. It kills all the rest of them. So you can have both. We talked about it. It can be selective and differential. Inoculations. Okay, we did that just by what we did before with our differential media. You can see how you inoculate it and actually form words. You just put it on your little tip and draw like that. And then we got miscellaneous media. It's like a carbohydrate fermentation media. It allows growth with fermentation. Contains sugars. Okay. That can be fermented. Sugar. The breakdown of sugar is fermentation. How we make wine, how we make bourbon, vodka, any of our alcoholic drinks. We ferment. We can ferment grapes. Okay. They break down the sugars in it to CO2. As it breaks down, it forms the alcohol. Converted by fermentation, converts to an acid, and the pH indicator is show the reaction. So as it breaks down, it releases H+, it releases CO2. That's what gives you the bubbles in champagne or the bubbles in your drinks. So you can see the gas bubbles that are being formed from fermentation. And this is the tube that's inside this media inside it, and it's growing inside this tube. You see the cloudiness here? That's indicating growth of your microbe. And then it's forming gas, and the gas is rising up at the top of the tube.